All right, we can get started. And so this is where we're at. We have our third exam this Thursday, and we know what it covers, we know what the format will be like, and we can ask questions all day today. Then after exam 3, there are not going to be any more assignments or assigned reading, but we'll cover important additional material and next week. And if there are eliminator questions today, I will start talking about stuff that will be on our final but not on our third exam. And then the final is the same style as our other exams, covers the entire course, and it's on Tuesday the 5th at 8 a.m. in here. So we're talking about this, and that's a big focus of our sort of cell biology oriented third exam. So let's check in with people to see if people have questions about anything at all.
Questions about the figure?
Anything. Yep. So we're getting ready for our exam, and if you have questions, anything about what's going to be covered on our third exam, we should talk about it today. Yep.
Is lipid raft the same thing as the fluid model, like mosaic model?
Yeah, no. All the cell membranes on Earth are composed of phospholipid bilayer, and then they have proteins in that phospholipid bilayer, and it's very fluid. So those are diffusing side to side very fast in general, both the phospholipids and the proteins. So that was the original term for a cell membrane, was this fluid mosaic model. So fluid moving around a lot. Even though it's a barrier, the molecules are moving around a lot. And then a mosaic is kind of an artistic style, certain type of art pattern. So it's a mosaic, the mosaic being these different functional proteins in the membrane. And so that was introduced in 1970 about with this idea. And scientists based that on a lot of different evidence that that was the structure of every cell membrane on Earth. And the membranes that surround organelles like mitochondria and so forth, that phospholipid bilayer is also a fluid mosaic model. So we start out with that. We're like, oh my gosh, those things are moving around pretty chaotically. And then it was relatively recently that scientists found these islands called lipid rafts. And they're composed also of lipids and proteins, but there's a higher concentration of cholesterol and sphingolipids and hydrogenated phospholipids, so that the lipid raft is what it sounds like. There isn't as much movement, it's not as fluid. And so it provides an opportunity to have organized structures of specific proteins contained there. And so the overall cell membrane is doing a very sophisticated job a lot of different ways. Lipid rafts provide a kind of localized organization to do some specific tasks, and there are lots of different ones. The list goes on and on. And we really just mentioned one. Our example is that extra amounts of LDL receptors get stored in lipid rafts. So normally the LDL receptor is involved in receptor-mediated endocytosis of LDL, and if a cell just has plenty of cholesterol, then it will hold that extra LDL in the lipid rafts like that. And just to add another kind of memorable idea to that, so we learned that lipid rafts include higher levels of cholesterol, that cholesterol makes this the membrane stiffer, more like margarine and less like vegetable oil. And if there's a high level of cholesterol, you would want a reduced intake of LDL. So there's a natural relationship between, in one cell, higher level of cholesterol, more lipid rafts, stronger lipid rafts, more holding of LDL receptor, and reducing the intake through receptor-mediated endocytosis. And so there's this natural kind of feedback regulation of the amount of cholesterol in a cell that way. There are lots of other mechanisms. And again, lots and lots and lots of things that lipid rafts do. But that's our one highlighted example. And in our list, let me just go to that. You know, in our list, each of these examples have, lots and lots of things they do. But we picked one example to, really to be able to focus in on and be like, oh my gosh, the aquaporin and urine production and how you adjust the volume of urine. Neurotransmitters in your brain, all the time you're thinking, just massive amounts of exocytosis and endocytosis of the neurotransmitters, but also the cell membrane. The cell membrane, when the vesicles fuse with it, cell membrane gets bigger. And so we need to have endocytosis, both to just have that lipid traffic going constantly, but also to help pull back neurotransmitters into the presynaptic neuron. So a really famous example of just tremendous amounts of exocytosis and endocytosis going on in our brain. And then our third example was this, kind of an extended example of the LDL receptor and receptor-mediated endocytosis, and bringing LDL in and its relationship with heart disease. And then we had the lipid rafts, and then our last one is just a very, another relatively brief example based on all the things that cell engineered molecules do, with one example, and that is when there's an infection, a local infection like from a wound on your finger, and macrophages have to leave the blood vessels, crawl out into the tissue. They do that by landing and rolling or crawling to the specific site, and then squeezing in between that tight barrier that the endothelial cells make to keep our blood in our blood vessels. So, nice, memorable examples of very important things. What chapter 14, cell membrane activities like that. Yep. Yeah. Other questions? Anything at all people have? And we can, reach into the final exam also, right? We'll be doing that next week too. But like I mentioned, I don't try to put together a summary review. I'll just roll on with stuff and slow down when somebody says, "What about that?" Yep. Yep. Yeah, Sophie.
You talked about post-translational modification and regulation. How would we differentiate what is grouped in each?
Yep. So the big category is post-translational regulation, and that means after a protein gets synthesized, is its activity controlled. And then the smaller set of that is, in some cases, post-translational regulation includes covalent modification to the protein. Any covalent change to the protein is called a modification. And those are nice words, they're post-translational, right? After the protein gets made, we can still regulate its activity. So the regulation, any example, right? And then modification, physically altering the protein, is a subset of the two. And then we can think of examples. So for example, the FOXO being bound by 1433, that's a post-translational regulation. We're turning it off, turning the activity off, but not by modifying the protein. FOXO, in that same example, don't let it get complicated, but FOXO has also, the way that 1433 decides to bind to it is that FOXO goes through a post-translational modification. It gets phosphorylated. So FOXO has a post-translational modification, phosphorylation, that leads to a post-translational regulation. And so the terms are nice, they fit together, and we just have lots of examples. And remember to go back to the idea of our protein transcription factor list that we made for the first exam. Those are all examples of post-translational regulation, including some that are modifications. So the Notch receptor gets cut. So when the protein gets cut, that's a post-translational modification, right? Because it's changing the protein. And throughout our course, we ended up with several examples of proteases cutting protein and changing its activity, right? If a protein gets cut, that's a modification. And then We talked about that muscle differentiation, and we brought up some other post-translational modifications, that samoylation and glycosylation and so forth like that. And we added it to our muscle story because our muscle story is a good one, and we should be able to add some more kind of complicating details to it, right? It's all of these things we talked about in our course are not oversimplifications. They're just where we zoom in and try to get a handle on an important principle and concept. And then, over the, time, you want to gently zoom out a little bit and add a little bit to that. So we did that with that muscle differentiation example, just to make that post-translational modification list a little bit longer. Like that, yep. We talked about ubiquitination, like that, leading to really typically degradation. Yeah. Yep. Yeah. Spencer, do you have something?
Yeah. Regarding the modifications during muscle differentiation, would you ask us specifically about one, or would it be like list to describe like 2 examples of?
Yeah. Yeah. Like that. We were just trying to make a list there, just to kind of reach into that and show, oh well, we really learned a lot of examples of phosphorylation. But there are really comparable modifications where you're adding a group, glycosylating, samoylating it, ubiquitinating it, and so forth, just to expand our awareness to that list, and but not dive into the detailed examples from that. Yep. Yep.
The glycosylation in the Golgi lysosomal proton pump, I drew out the pathway leading to the mannose-6-phosphate. Is that the key takeaway from that? Because we drew something, the cell membrane with the PDGF receptor.
Yep.
I don't know.
Yeah, let's draw that out. Yep. So we could start out with that list again from the assignment, where we say, okay, all these proteins are going to get synthesized initially starting on a free ribosome. So free ribosome was just floating around in the cytoplasm. There are tens of thousands of ribosomes in each of our cells, and it binds to an mRNA. And this time the ribosome happened to bind to hemoglobin or any other protein that is going to get synthesized and just released into the cytoplasm. And that's what happens to hemoglobin. The hemoglobin in our red blood cells is just in the cytoplasm, floating around in the cytoplasm. But there's many thousands and thousands of proteins that fit that category. They're simply synthesized by free ribosome and released. All the proteins that have to get into the nucleus are synthesized by free ribosomes, released, and then they got to have a nuclear localization signal to get into the nucleus. All the enzymes in glycolysis and so many other things are the cytoskeleton. Those proteins are just synthesized and released. So thousands and thousands of proteins fit that example. Then another massive number and fit this where they need to get to a specific destination. And so on our list in the assignment, we had insulin, it's got to leave the cell. We had PDGF receptor, got to be in the cell membrane. We have the lysosomal proton pump that Spencer asked about, so we'll get to that. But it's just nice to see that. And there are exceptions to proteins, say, leaving the cell. But there are few. Most of them, all of them, start out being synthesized initially by a free ribosome, and the beginning of the coding region of those proteins has about a 20 amino acid portion immediately called the signal sequence. And the free ribosome starts to synthesize those proteins, and as soon as that signal sequence starts to emerge from the ribosome, that's the beginning of this protein, the ribosome pauses synthesis because a signal recognition particle, SRP, binds to the signal sequence and says, you should pause. And it just pauses until the ribosome floats around and bumps into the rough ER, binds to docks on the rough ER, and then it resumes translation. And so that's where lots of protein synthesis takes place. That's the number one function of the rough ER. The rough part of it is it's dotted with ribosomes, and that can be seen under electron microscope. It's the rough ER. It's the site of protein synthesis that needs to go to certain locations. So insulin simply gets synthesized and released into the lumen of the rough ER. and then it's going to pinch off in a vesicle. The newly synthesized insulins will be floating around in the lumen of that vesicle. The vesicle will fuse with the Golgi, it'll go through the Golgi, and then more vesicles will take it to the cell surface and exocytosis. And upon exocytosis, the insulin just gets released outside of the cell, in this case, into the blood. So proteins that are secreted proteins go through that pathway with very few exceptions. Great. Then PD receptor, really similar story. It's just that it's a membrane protein, a transmembrane protein. So it also initially is synthesized in the rough ER, and it gets inserted into the phospholipid bilayer membrane of the rough ER. And it gets inserted in a certain orientation because it's just going to be in a membrane. It's got this hydrophilic waist around it, like a belt, and it just needs to be bobbing in a phospholipid membrane all the time. And so that also is going to pinch off as a vesicle, go through the Golgi, go blah, 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 out the cell like that, or to the cell surface. When secreted proteins or membrane-bound proteins go to the cell surface, the phospholipids end up there too. So when we do exocytosis, we're adding to the membrane. There's got to be membrane traffic, happening. We're going to have endocytosis, just bringing phospholipids back into the cell, sending them back to the Golgi and the rough ER, right? They got to keep a balance of phospholipids in them. So there's just a lot of membrane traffic, a lot of vesicles moving around like that. Okay, so then the last one. The last one is this lysosomal proton pump. And I think I have some slides just because I thought that might come up, and we'll add a little bit to our story. So let me just zoom down to those. And these are things we've generally talked about, but we can roll through them again, and they're, those examples. And I think it was Sophie that asked about what do we need to know about any diseases. So let's just talk about the lysosomal proton pump. Let's learn about 2 diseases because it'll help us, oh, I got that, right? So one of them is called a lysosomal storage disease. It's actually a group of diseases. They're inherited because they involve mutations in genes that code for proteins that are important in this pathway, this special pathway the lysosomal proton pump is going to go on. And the other one is called eye cell disease. And so we'll just learn about those. And they involve, a child that's born with, inherited problem. The problem is they have a mutation. And we're going to talk about 2 different proteins that are involved in the lysosomal proton pump traffic and how these are loss of function mutations. We aren't going to get the lysosome really built correctly. And the phenotypes for these is the babies. If you look at their cells under a microscope, you see they have really big lysosomes because the lysosomes are not functioning well. And they're not functioning well because the lysosomes are like the cell's stomach. They're acidic. They're supposed to be breaking down molecules. And these lysosomes do not have an acidic lumen because they didn't get the lysosomal proton pump delivered. And so it's just the trash built up. They can't get rid of it. And it's a very tough disease. Now, next thing to mention about that is, we mentioned this before, there are alleles, right? So how badly damaged are the proteins that the child is making, right? Because there could be a child born who has, somewhat reduced, and they're okay. They don't have, it's working some, but not, not as well as it needs to work, okay, like that. So there'd be a range in the phenotype of these diseases. Okay, so, and we're just gonna add this because it'll be a nice thing, okay? So now let's just go to a picture, and the picture looks like this, where we're talking about that lysosomal proton pump, and we've got the rough ER, and the Golgi, and The Golgi is where our lysosomal proton pump story begins. So it's going to get synthesized in the, I'll change the color for this, we'll say. So going to get synthesized in the rough ER, going to go by vesicles to the Golgi. It's going to be in the Golgi. And in the Golgi, the lysosomal proton pump is going to get glycosylated. So it's going to be a post-translational modification. It's going to get glycosylated. And that means it's going to be a glycoprotein, a protein that has the protein portion of it, and then polysaccharide sugar on it. And those can be complicated, have lots and lots of different sugar molecules. And the one that's relevant here is a sugar. It's not, glucose, sucrose, fructose. It's a sugar called mannose. And that's happening in the Golgi. So the lysosomal proton pump proteins can be identified in the Golgi, and the Golgi says, we have to help direct you to the lysosome. So we're going to be careful. We're not going to send you to the cell membrane. We've got a lot of proteins coming through the Golgi, and they're going to the cell membrane. This one's going to have to take a turn, and we'll talk about it. It's going to take a turn here to go to the lysosome. But before it goes to that point, it's getting modified in the Golgi. And so that's where our first enzyme is that we want to learn about. And the enzyme that is going to add a phosphate to make that, it's called mannose 6-phosphate. So that's the final tag we need glycoprotein with phosphorylated mannose. And we're not going to learn the name of that disease because it has a long name, the enzyme, but we're going to learn the name of the disease. If a person has mutations in that enzyme, they're going to have this eye cell disease. And the eye cell disease is caused by an enzyme. It must be a kinase that adds that phosphate. And you've got to have that phosphate added. It's not enough to have mannose as a glycoprotein. You've got to have that phosphate added, okay? And so that's what is happening there. And then, vesicles are going to be released, and I'm not doing a great job with my color coding, but here's some more vesicles. And here's where we're going to take a turn like that, okay? And so this endosome, this vesicle, scientists like to call these vesicles in this region, they're seem to be heading to the cell membrane. They call these endosomes. So it's going to be an endosome where we find the second protein, and the second protein, I might have to write up in a different location here, but the second protein is in this endosome, and it's called the mannose 6-phosphate receptor. And let's just think about that. Just pause our thinking about that. So this mannose 6-phosphate receptor, well, it's a membrane-bound receptor, but its job is not really in the cell membrane. Its job is in this endosome. So this mannose 6-phosphate receptor is staying here. So we're going to see this mannose 6-phosphate get recycled or reused. It's going to grab some lysosomal proton pump and other lysosomal proteins, bring them here, and then drop them off, and then return. And so it's a really satisfying picture. Just in this region of the cell, there are vesicles that are just going like that, a little treadmill of just hauling mannose 6-phosphate receptor, looping around. And the mannose 6-phosphate receptor, we can learn the name of that because it's a sweet name, you have to admit, right? And if you have a mutation, if the person hasn't been born with a mutation in it, it's an example of one of these lysosomal storage diseases. And there are other causes of them, but another sweet name, right? You know, it's like, okay, yeah. If you have a mutation in this mannose-6-phosphate receptor, it fails to grab the lysosomal proton pump's mannose-6-phosphate, fails to get it over there where it needs to go to, like that, right? So it's nice to kind of dive into a little bit. There are other parts of the story, other types of lysosomal storage diseases like that. Just to say it, we're focusing on one protein. There are other proteins that need to get here. There are other lysosomal proteins. There are enzymes in the lysosome lumen that need to chew it up, you know, chew up stuff like that. It's not just acidic, but enzymes that are going to break stuff down too. And those are proteins, and they need to take this same pathway. So there are several proteins that have to have the mannose-6-phosphate as glycoproteins, and they get grabbed and brought over to the lysosome. I mentioned in class that there actually is a little bit of mannose-6-phosphate receptor on the cell surface, because if you accidentally get some of these to the cell surface, you're like, grab those and get them back in here, because that's the wrong place. We made a wrong choice. That's just a minor detail, but it's kind of cute to see it, you know, in action like that, okay? And, but so locations, right? Cell biology, nice locations, right? One story is here, right? Got to get it modified, got to get ready, and then the next story is here in the endosome. Ah, we see you. We're going to take you over there and just be doing our little loop there, yeah? So that lysosomal proton pump gets synthesized here, and that's its pathway. Heading toward the cell membrane, and at the last minute, it's like, whoop, nope, the mannose-6-phosphate receptor grabs you and keeps you in this endosome to lysosome traffic pathway, yeah? And there are more details, but they're more of the same. You know, it's all about, you know, modification of the Golgi, and then in these endosomes that people call them late endosomes. But yeah, Liana?
So, the receptor is going into the lysosome and then being recycled into the endosome?
That's a good, yes. So, right, because we could, you know, draw these vesicles. The vesicles are going to fuse with the lysosome. And so, yeah, the mannose-6-phosphate receptor is in an endosome, then it pinches off and it's in the membrane of a vesicle, and then it does fuse here, so it's temporarily in the lysosomal membrane, and then it has to get, you know, taken back. So, you know, a vesicle has to pinch off from the lysosome that has lots of mannose-6-phosphate receptor that has delivered its cargo, and now we're taking it back. Yep. Yeah, you know, and people are like, wow, you know, life is complicated, and you're like, oh man, oh my gosh, tell me about it. Yeah, and, but you know, this is just the satisfying thing to see, you know, these proteins in the lysosome are really important, and you know, you could have a mutation in the lysosomal proton pump, or a mutation in any of the proteins needed to get it where it needs to go. And you're in trouble with, you know, some defective lysosome. Yep. Yeah, so we should be able to briefly describe that or draw and label where are these. Yep, and we just added that detail again, not knowing the name of that enzyme, but the reality is you got to add a phosphate to the mannose. And our book has a figure, and I don't think our book names the enzyme, but our book shows this, and see it shows that it's getting glycosylated in the Golgi lysosomal protein, and it's getting modified, and ultimately it gets that mannose-6-phosphate ready, and the enzyme added it there, and mutations in the mannose-6-phosphate receptor are the next one, and that's shown here a little bit. And these sort of blue, you don't need to see this in detail, but the blue like Y's are the mannose-6-phosphate receptor, and you can see it's hanging out here, and it's delivering, you know, going from the Golgi into this endosome, and then it's getting recycled back like that, back and forth. Oh, and it looks like before it actually, at least from this drawing, it maybe never fully fuses with the lysosome. Maybe it gets recycled back before the delivery, but that's a minor detail. Yep. The idea is this is where the mannose-6-phosphate receptor is doing its job, spending its time in that little looping pathway. Yep. So, we've just taken the, nice buildup, hemoglobin for ribosomes. Then insulin, rough ER, it's gonna get, released from the cell. Great example. All those cell membrane proteins, PDS receptor and so forth, same thing. Really similar pathway, they're just membrane proteins. Then the last one, lysosomal proton pump, we gotta do some special work to get it over to the lysosome. And for the, on the exam, I could switch and say, instead of talking about PGF receptor, I could say, well, what about the, any other cell membrane protein? What about aquaporin? How's that get synthesized? And you're like, well, I bet it has a signal sequence, and it starts out getting made by a free ribosome, then it has to dock in the rough ER, and newly made aquaporin is in the rough ER, and it's got to get to the cell membrane, Because these are specific examples to give us general rules for thousands and thousands of proteins, almost all the proteins. Not all, there are some that are made in the mitochondria or the chloroplast and so forth, but the vast majority of proteins like that, yep. And that's a big part of chapter 11 also, yep. I think that's all that's going to have of that, yeah. Other questions, more on that or anything else? Yep.
So I just wanted to clarify the orientation of some of the proteins being processed. So if a protein is synthesized in the lumen of the rough ER, like insulin, then it is on the cell membrane, then it's on the cytoplasmic side to then be released into the bloodstream. But then like RAS is synthesized in lumen to be docked or anchored to the
Yeah.
cytoplasmic side.
Yeah. So let's look at the rough ER. I'm good. Slides. So let's look at the rough ER membrane and get that orientation. And so we'll draw, here's the rough ER like this, and this is going to be rough ER. And we'll just add some color to the phospholipid bilayer. So see, this is that two sides of the phospholipid bilayer, just to start with, to get that orientation. And when those phospholipids make it all the way to the cell membrane, so they're going to pinch off in vesicles, then they're going to fuse the Golgi, pinch off in vesicles, eventually exocytose to the cell membrane. We're going to see that this will give us our orientation, that those phospholipids will now be on the outside of the cell, because it's like when it fuses, it joins that surface that way. And those aqua-colored ones are going to be on the inner membrane. So just kind of using that as a reference point, we get this, where that means that insulin here in the lumen, is going to be insulin that gets released, because that's that same side of it. And so it essentially the liquid interior of the rough ER and the Golgi and the vesicles all are going to be equivalent to the outside of the cell, like that. Similarly, they would be comparable to the inside of the lysosome. You draw that out too. Then we have Priscilla also asked about RAS, And so we can add RAS to this and say that RAS then is going to be made, it gets made by ribosomes on the rough ER, and it's going to get anchored to this side of the membrane, so that we'll see RAS on the inside surface, the inner leaflet, anchored to the inner leaflet of the cell membrane. And the orientation is just always that way. There's no way to switch it around like that. Now, it's, that's our story with the those proteins, but let's just go over here and remember that the smooth ER is synthesizing lipids and fats, and the smooth ER also has that same orientation. So these phospholipids are equivalent to eventually being the outer side of the membrane, and these the inner. So we want, say, something like phosphatidylserine, that apoptosis lipid. We want that to be on this side, and this is the side where almost all the synthesis takes place. We also want that HIP3 on this side also. On the other hand, we want some other phospholipids on the outside, and so we do have these flipase enzymes that can flip proteins from side to side, or phospholipids from side to side in the smooth ER to get them orientated. I shouldn't have said proteins, I should have limited it to smooth, to lipids and phospholipids. Yep. So that's also the same orientation sort of story. And that orientation story is very physical, drawn out, and you're like, yep. Or literally build it and join these two together, and that's where they're going to be, on that side or on that side, always. Yep. Yeah.
Our book talks about cystic fibrosis. Should we know about that?
No, I'll say no. Yep. So cystic fibrosis is a mutation in a gene that codes for a protein that's a cell membrane protein and is involved in ions passing across the cell membrane. And our book talks about ion channels a fair amount in that chapter, and I didn't really talk about them at all in class. And maybe there would be a question on the exam from that, but probably not. And the reason I didn't talk about it very much, the membrane polarity and ion concentration differences across membranes are really important. But Doug Curry teaches neurobiology, and then every other spring he teaches animal physiology, and he really likes to talk about those stories. And so I figure, either students will take those classes, or we just won't put a lot of energy into it here. Yep. Yeah. Yep.
What
Kayla?
about like the 3C techniques in the fluorescence?
The what?
The 3C techniques in the book?
Oh, I don't remember what those are. Do you have a page, or do you? 3C?
Techniques, yeah, and like the fluorescence and SDU. I think it was chapter.
Yeah, I don't know. I can't remember what those are. Yeah, does anybody else have those down? Yep. I guess they're not going to be on the exam, I can say that much. But it's 3C? Yeah, I don't remember what that is. Yeah. Yep. Do you remember, is there a context to them, like what they're being used for?
I think it was like something about like the channels. I can't remember.
Okay. Yeah. I'll try to find it, and it won't be on our exam, but I'll look it up because you asked about it, and try to follow up next week. And either say, oh yeah, that's something really cool, we should learn about it, or say, oh yeah, that's kind of an add-on. Yep. Yep.
So do you want us to be able to like draw our cycling of the RAN/GTP?
Very
Just the
limited, right?
important.
Very limited examples of that. Our focus is really on important and exporting, and the story would be that important is going to be bringing cargo into the nucleus, exporting, taking it out. And for the important, really emphasize that cargo has to have an exposed nuclear localization signal to bind to important, and then important comes in, then important has to leave. So we also would, on a very simple diagram, have information to show that it's got to actually involve breaking a GTP. That GTP is bound to that RAN protein, so that is necessary to get it out. But you wouldn't even need to name the RAN. Just say that in the inside of the nucleus is where there's an energy dependent step to reuse to get important back out. And then same story for exporting. We didn't even mention, but molecules need an export tag to get out that export can bind to. It too also has to burn a GTP in its cycle in order to keep the cycle going in one direction. Yep. But yeah, there's a lot of detail in the figures in the book, and we don't have to dive into those. Yep. Yep.
With RNA splicing, oops, the SNRP like splices introns out to produce the mature transcript of exons. Is that true?
Yeah. Yeah. So let's just draw that and to make sure everybody's got a handle on that.
Can I have another question kind of along with that?
Sure.
The isoforms, are those the result of alternative splicing of just exons, or can they be introns as well?
The answer a person would say is it's really exons, but I'll draw that out to a separate one. So the first thing to say is we're talking about this RNA processing. It takes place inside the nucleus, right? So we go to a location like that. It's going to happen inside the nucleus, and we first have DNA, and we draw out a lot of times. We draw out that this is the transcription unit. And I've never read this, but I'm pretty sure this is true. There's only one way to output information from DNA. So DNA is the primary heritable molecule on Earth. It's an information storage molecule. I think the only way to get information out of it is to transcribe the DNA into RNA. And the next thing you'd say is, like with a computer, you don't open all the files, you open them as needed. So a transcription unit is literally a discrete portion of the genome that we want to output. And we're going to output it by transcribing it into RNA using the RNA polymerase. So we're going to get a primary transcript out, and the primary transcript is exactly the same length as the transcription unit, because that's the definition of the transcription unit, is that's the portion that's going to get transcribed. So we have those exactly the same, and then ultimately we're going to have RNA processing in the nucleus, and then what leaves the nucleus is going to be mature RNA. And that's true for coding genes, and the mature RNA would be messenger RNA. That's true for non-coding genes, is they're going to go through this RNA processing, and the mature RNA leaves the nucleus. For this, we'll focus on the coding genes and their modifications. And so we have this primary transcript, and there's a first round of labeling we'd want to do. We'd want to say that we know that if this is a coding gene, that's the coding region. And the coding region is not as long as the primary transcript. So this is the coding region in the primary transcript. This is the coding region in the mature transcript. So the RNA is longer, and so we got a couple labels to add, and that is, we have this region, nicely named 5', it's the 5' end of the gene, 5' untranslated region. And then this is the 3' untranslated region. And it's just, they are what they say they are. They're just extra pieces that are there. And sometimes there's regulatory information in them like that, and they vary in length. Then the next thing we want to say in this first drawing is that we also want to mention that we have these modifications that occur to the primary transcript. And so we have this 5' cap added, and that would be here too. And the 5' cap basically helps stabilize the RNA. And then we're also going to get a poly A tail added after transcription, and that also helps to stabilize the RNA. Those modifications are also essential for translation. The ribosome recognizes these portions, so we gotta have it for stability and also translation. So that's our first drawing, and for some genes, it's as simple as that. But in some cases, there are portions of the transcript that are going to get removed, and those portions are called introns. And we're going to just draw a real simple example of one where there's just going to be one. No, we'll draw a simple example where there are going to be 2 introns. So all these are true for the next drawing. We just have to kind of switch so our drawings don't get too complicated. But it's also true for these drawings, okay? The next drawing, okay? So we go back to this and we say, oh, yep, there's our DNA and our transcription unit, and here's our primary transcript. And then here's our mature transcript, shorter. And the mature transcript is shorter because of RNA splicing, which is an example of RNA processing. And in the RNA splicing, the introns are removed. So if the gene has some introns, they're in the transcription unit, and they get spliced out as RNA, and the mature RNA is shorter than the primary transcript. So if there are introns, we can see them in the DNA. So they're present in the DNA. There's an intron, there's an intron, and they'd be here also, and there would be no introns there. The introns get spliced out. And the introns define where the exons are. So this one would have 3 exons, exon 1, exon 2, and exon 3. And, you know, where's the coding region? Well, the coding region is going to be in those exons, and it varies. Maybe some of the coding region would be in exon 1, but maybe none of it would be. So, again, it's there, but it's just a different drawing that we have to shift to. Okay? And so, if we go out to the mature RNA, we would say there's exon, exon, exon. The mature RNA is composed of just exons, and it's the 3 exons. So now let's just pick one of these introns and draw that to show what a splicing looks like. So here's this, just one intron showing that, and we're going to splice it out. And the splicing is done by some ribonuclear particles called SNRPs, and they're going to bind here, and there are multiple SNRPs. And they are involved in that splicing process. Okay? And so we want to just talk a little bit about the SNRPs. So this is the molecule that's getting spliced is going to be on a coding, it's a coding gene. And so that's mRNA. SNRPs are small nuclear ribonuclear particles. As part of their function, they include s and RNAs. which are examples of non-coding RNAs. So they're not coding genes. There are snRNA genes that code for these small nuclear RNAs, and they're involved in that splicing. So it's important to appreciate that in a SNRP, we have both proteins and RNAs. And let's see, and you had that in that context of, did you have a detailed question about that, or just to show that SNRPs are involved in splicing?
Yeah, I just was looking for clarification. to make
Yeah.
sure that was.
So that's the function of SNRPs, is they're involved in splicing. And so we could draw a little picture of that, or just say that, where do SNRPs function in the nucleus? What's their function? RNA processing, what do? Splice out introns. Okay, so that's Spencer's first question. And in the chapter, in our book that talks about, exporting and importing things into the nucleus, it dives into SNRPs because they have kind of a complicated story of how they get put together in the cytoplasm and then get imported back into the nucleus, and we don't have to go there. We would just say, let's look at some already functioning SNRPs and where they're at and what are they doing. Yep. So one reason that people are interested in SNRPs is because this gene could go through alternative splicing. So this is one product that gene could produce, but another would be to make a RNA product that just is exon 1 and 2, or 1 and 3, or we'll say this is 1. And this is a regulated process, and it's called alternative splicing. Okay? And the SNRPs are responsible for reading that, and there are a bunch of different SNRPs and doing that. There's a lot of alternative splicing in us. And so then Spencer asked this kind of question about introns versus exons. And the intron exon is functionally defined, right? So we said here, oh, there are 2 introns, see them getting removed? But in that product, there was just one intron made up of introns 1, exon 2, and intron 2. So, right, so sometimes an exon, and that gets advanced and we don't want to go there because it's confusing, really. We just go like, exons get put together, introns get spliced out. But yeah, when you start saying, what about alternative splicing in introns versus exons? Then you have to be ready to kind of shift that around some. And there are just any number of different stories about alternative splicing. For example, the splice junction here could just be moved over some. And so it's still putting together the same thing, but just not as much of that exon. So lots of complicated things, so lots of enthusiasm for SNRPs like that. Yep. And that's as far as we have to go with that. Then next thing is that because of alternative splicing, we could also make more than one protein from a gene. Example of that would be the estrogen receptor. The estrogen receptor gene and testosterone receptor get alternatively spliced. So they make multiple protein products. The protein products are like this. Parts of the proteins are exactly the same, and they're just missing or adding other parts. Right? So let's say these were all part of the coding region, and this would be like, a head, body, and legs, and here's just head and legs. The head and legs are identical, just missing one part. Alternative splicing could mix and match them too. You could have 2 different body exons and mix and match them like that. So lots of variety, but all being made from the same gene, and there are parts of the proteins that are common, identical regions, and parts that are different or missing, like that, okay? So those proteins from one gene that have been alternatively spliced, those are called isoforms for the protein isoforms. And the key concept is they all came from the same gene, but they are, you know, different, and they could have some slightly different activities like that, you know? So those are isoforms. Now, we're gonna be careful to distinguish those from paralogs. So that's not, these are not paralogs. Paralogs are proteins that are made from different genes, and the genes arose by gene duplication. So ancestrally, the genes were identical, but they become different over time. So paralogs have also some regions of identical, but also just differences along the length of the protein. Could be anywhere in variations. So the proteins look like kind of a similar story, but they're distinct. And the simple distinction is paralogs come from multiple genes, isoforms come from one gene. Yep. And it's about the proteins and the protein product and how it functions. Yep. Yep, and I think that covers the stuff you brought up. Yeah, other things people want to talk about? Yeah, Chase?
With the 3C method, I remember now was like one of the 2 experimental methods in like the nucleus chapter, like talking about chromosome location.
Oh, okay.
The other one was fish.
Yep.
Do those sound familiar?
Yeah.
I didn't write about them too detailed in my notes on the chapter.
Yeah. So fish is fluorescence in situ hybridization, and I guess the 3C, which I still can't remember, is probably doing something comparable to that. And it's a way of visualizing specific genes or parts of chromosomes within the nucleus. So it goes to that thing where we're like, oh, during interphase, the chromosomes are relatively decondensed and just look like a smooth gray area. But there are specific regions that we just learned about where we have some transcriptional activity, you know, localized. Yep.
The Center Chromosome Conformation Capture for 3C, it like explores like the association between chromosomal regions and like different chromosomes and like cross-linking of different sequences.
Oh, yeah, okay, yep. Yeah, so fitting into that same thing of wonder where those chromosomes are. If we care about chromosomes, can't see them. Do they have any organization? And they do, but the organization is not yet, I would say, like fantastically awesome. Like we're like, dang, you know, that's really nice, right? Like, you know, we talked about the lysosomal storage disease, and a student can be like, okay, all right, but there aren't any really great take-home messages. There are probably, but the organization of the nucleus is happening. What we really focused on there was just the statement of transcriptionally active genes tend to be near the nucleolus or near the outer nuclear membrane, and then we talked about some nuclear bodies. So one nuclear body we talked about was a transcription factory like that, or a DNA replication factory. So that's where we would have got some of those statements like that. Yep. Yeah.
Back to the RNA splicing, you
Yep.
said that RNA processing happens for non-coding and coding genes. Is RNA splicing only for coding?
Yeah, that's a good question, and I'm going to say yes. I bet there's exceptions to it, but we would lean towards saying yeah. That's it. The 5' cap, the poly A tail are a big time coding gene, mRNA modification. Splicing too. I don't know of splicing in non-coding genes. Non-coding genes, the RNA is going to be the functional product. And so there we talked a little bit about the RNA processing of non-coding genes. The RNA of non-coding genes often gets some covalent modifications to it. Like the book shows it a little image of tRNA, transfer RNA, and it shows all kinds of covalent modifications. So that would be examples of RNA modification of non-coding genes. And it's satisfying because that's going to be the functional product. So let's make, there'd be natural selection pressure to make a fancy functional non-coding gene product. And another one that the book talks a little bit about is RNA editing. So in some cases, non-coding RNAs and even some coding RNAs have one of the nucleotides gets changed. And those would be covalent modifications. But for us, The big story is what we drew out, those would be really dominated in the stories of coding genes. And then when we turn our minds to non-coding genes, we would want to say that they have to be modified to produce the mature functional product. And sometimes that involves a modification by adding, simple groups like methyl groups or acetyl groups. We talked some about RNA interference and microRNAs and small interferon RNAs. Those are non-coding gene RNAs, and those get RNA processed. They get cut to make a really short final product complex like that. And we just didn't go down that pathway very much about non-coding genes. Yep. Yep.
How will we draw endothelial cells being held together by adherence?
Yep. Yeah. So for our example about cell adhesion molecules, we said let's learn about 3 classes, selectins and CAMs and cadherins. And our attention is on the macrophage in the blood. So this immune system cell, white blood cell called a macrophage, that loves to phagocytize bacteria. And it's going to it's just right now, it's just floating in the blood, getting pulled along with the circulation, but it's detecting an infection in this tissue. And so it needs to leave the blood. And so, that's the cell that's the cell we're paying attention to. But like Dornika asked, this is where the cells are expressing those cell adhesion molecules that we want to learn about. And it's the lining of the blood, and these are endothelial cells that line the inner surface of all our blood vessels. And so it's those cells that are expressing these proteins, and they have these proteins on their cell surface. Cell adhesion molecules are cell membrane glycoproteins, and we just have so we'll say all 3 of them are present all the time, like that, on these cells. And then we'll say, okay, well, so the grays are the cadherins, and the purples could be the selectins, and the orange can be the CAMs. Like that. So they're always there on the endothelial cells, just on the cell surface, and they're binding sites for proteins here. And these proteins here, I should do a different color. These proteins are different proteins that are going to bind, that are on the macrophage. And the macrophage is first going to be connection with the selectins, and then it's going to make, it's going to bind here, there, and pause, not be rushing along with the blood, but pause. And then it's going to crawl and roll along these CAMs, little handholds like that, like you're a rock climber and you're climbing along the surface. And then the macrophage is going to get to this place, and it's going to release some protease enzymes to chew these up temporarily so it can squeeze between those cells. So that's what it's doing there. That's where you asked about the drawing. You would draw, 2 endothelial cells holding hands with these cadherins, and the macrophage needs to get between them. So it's just going to temporarily destroy those proteins, squeeze through, and the endothelial cells are constantly making more of these molecules, so they'll hold hands again soon. Yep. Yep.
And the adherence junction is between.
Yes, and this is the adherence junction. Yep. Yeah, the cadherins are what are making this adherence junction. And there is more to an adherence junction than that, but that's the center piece of it. Yes. And that's that tight junction that makes a seal, so the blood stays in, the blood liquid stays in the blood vessel. It functions, it would be a little bit like, you might think it a little bit like a turnstile. You know, when you're going into like an event and you go through a turnstile, you squeeze through there, but then it closes behind you again. Yep. Let's see, did Sophie have a question? Yeah?
Yeah, our book talks a lot about autophagy and autophagosomes. Like it goes into a lot of detail about it from like 404 to 408.
Yeah.
What should we take away from that?
Just that that the autophagosome is an organelle, and it's made up of a lipid bilayer, and when a cell is not going through autophagy, the autophagosome organelle is not present, and it gets assembled by some existing membranes inside the cell to make this autophagosome. And then the autophagosome does what it sounds like it does. It will just wrap around parts of the cytoplasm and phagocytize those parts. I mentioned we have, tens of thousands of ribosomes. Well, lots of times the cell's like, I don't really need that many ribosomes. So autophagosome just kind of magically appears in the cell, engulfs those, and then degrades, fuses with like a lysosome, and just breaks them all down and reuses, recycles all that. And that's the detail we would need to know. Yep.
It also talks
Yep.
about diseases with that, so we won't need to know
No.
that. Okay.
Yeah. Yeah, the diseases are almost, they're pretty typically an extra. And sometimes, the book is using them because it thinks it's a good example, but a lot of times that's just an add-on. But other times, like we talked today, sometimes they have nice names or nice functions where you're like, oh, I see what's happening there, and they can really be helpful. Yep. Yeah, other questions people have? And we're doing great. I think we're going to run out of time. And we have a few more minutes left, so we can have more questions if people have some. Yep.
Can you just go over like the disulfide bonding for after like protein synthesis? After the
Yep.
protein is formed?
Yeah. And so the amino acid cysteine has a sulfhydryl side chain that includes an SH group, and these can be separate like this, or they could join together in a disulfide bond, disulfide bridge. And it's really common in intro-bio to learn that, because these disulfide bonds often really help hold together the three-dimensional structure of a protein. They play an important role in the conformation and stability of the protein, like that. And but it turns out that, yeah.
That only happens in the
Right, and so it turns out that the chemical environment inside the cell, almost everywhere inside the cell, favors just having these free sulfhydryls. The extracellular environment really favors having these disulfide bonds. And so if you said, hey, how about these, that June Foss transcription, those transcription factors, I wonder if there's disulfide bonds in them. And the answer is no. They'd have cysteines, but they would be the SH group rather than a disulfide bond. And I just think, it's kind of surprising to hear that, because, intro-bio thing, you just start out thinking, oh yeah, it's probably important in various proteins in various locations, but it's not. So inside the cell, where we see these disulfide bonds is, like Kaylin mentioned, in the rough ER. That's where disulfide bonds form. And then they would be true for the Golgi and those vesicles that are heading out, that they have this environment that favors the disulfide bond linkage like that. Yep. And so, right, you could just have, like on an exam, it could be a list of proteins and say, which of these do you think have disulfide bonds? And you'd just be trying to identify proteins that are on the cell surface, or outside of the cell, versus those that are, you know, anywhere else inside the cell, outside of the ER and the Golgi. Yep. Yeah, but you know, saying a list of proteins, like, oh, you know, what about MIC? And you'd be like, nope, I don't think it has disulfide bonds, you know. And you can find rare instances of disulfide bonds of proteins in the, you know, intracellular environment. But those examples are like during an enzymatic process, there'll be a temporary formation of a disulfide bond and things like that. It's just very rare. Yep.
So would you see disulfide bonds on transmembrane proteins?
Yeah, and only on the extracellular part. Yep. Yeah. So PDGF receptor, you know, that's a great one because it's transmembrane, and there's a really good chance it has some disulfide bonds on the outside, but not on the inside. Yep.
Just to, I think you already answered this question, but, in the chapter about the nucleus, there's a whole section on the organization of chromatin. We didn't really go over that. Did you just want us to know mostly about like the replication factors?
Yeah, and then there is this, we did state this idea that if we look at we have those replication factories, and if we look that at the overall nucleus, so here's just the nucleus. And within the nucleus, there'll be this region that's the nucleolus. And people always, like me, always draw a circle around it, but it's not a membrane bound area. It's just an area of specialization. And so we'll put, I don't even know how to mark it, you know, kind of a dotted line. So that's the area of the nucleus. And scientists find that generally this is the region near the nuclear membrane and near the nucleolus where most of the transcriptionally active genes are located. And so if we said, where are the transcription factories, they would also tend to be, transcription factories would tend to be localized like that in those regions. And that's all we really want to get out of it. Again, you know, that's exciting. You know, interphase is when the cell does its, you know, living and working and functioning. So that's, you know, when genes are getting expressed and stuff, it's that interphase nucleus. And so, you know, scientists want to know what's the organization like. And there's some organization and, you know, some stories of regulation, but it's not a lot. Yep. And certainly not, you know, for like an introductory course, you know, it'd be an advanced topic where you'd be adding stuff on. Yep. So now we are out of time, and next week we'll have some new material like that, and it won't be we won't have assignments or reading assignments, you know, homework or reading assignments, but we'll be covering some new material that's important for our final. But we'll deal with that next week. This week we'll have an exam the next time we get together.